Hello, I'm Dr. Matthew Jorgensen, a board-certified toxicologist and senior chemist with Nielsen Laboratories. This is my poster presentation titled Unknowns, TICs, and Identified Compounds Below the TTC. One of the topics receiving a lot of attention in the E&L community has been the sensitivity required for extractable screening studies. This sensitivity is the analytical evaluation threshold and is based on the theoretically acceptable amount of individual compounds which might remain undetected in an extract. In other words, we need to know how small of a dose is so small that we don't worry about needing to identify it or report it. Usually, the analytical evaluation threshold is based off of a dose-based threshold, which is the threshold of toxicological concern. Now, the threshold of toxicological concern is an old idea. It comes from the ICHM-7, and this is an amount that was derived to be protective of unknown compounds that might be found in drug products or food products at low levels. You can see it's applied here to medical devices in ISO 21726. The amount that's acceptable as a threshold of toxicological concern depends on the duration of exposure. So for exposures that are less than 30 days, an amount of 120 micrograms per day is deemed to be acceptable. For longer durations, you can see that number trails down from 20 to 10, and then finally 1.5 micrograms per day for exposures that exceed 10 years to a lifetime. And this threshold of toxicological concern is directly applied in figuring out what the analytical evaluation threshold should be. You can see the formula here on this slide where you take the TTC, and because we know that there's some uncertainty in the semi-quantification during a screening process, we would divide that TTC by an uncertainty factor. And again, this is a topic that's very hot, and we could spend quite a lot of time talking about just how to compute this uncertainty factor. But for now, let's say that matter is settled, and we'll take an uncertainty factor of something like 4. So what we would say is that the AET is this TTC divided by a factor of 4, and this is the amount before you've gone through any of the effort of identifying or reporting anything. This is the amount above which you have to do the work to identify and report a compound. And what results is a situation like this, where you might have several peaks. You can see there's 8 peaks that would be reported here, and some of those peaks would be above the TTC, and others would be below the TTC but above the AET. And really the question is, for this brief time that we have discussing this together, is what really is the best thing to do, or how do we assess or evaluate those compounds that are above the AET but below the TTC? So after we've gone through this screening process, we might be able to identify all of those compounds. It's labeled as 3, 4, 5, and 6 on the graph there. And once we know what they are, we understand what the uncertainty related to their quantification is a lot better, and we can make now a toxicological risk assessment on those compounds. So one of the things that we've done in the past from a toxicological perspective is to say, the TTC is so conservative that if any compound observed is below the TTC, then we find that the risk of those compounds is acceptably low. Or in other words, if the TTC is protective of compounds even that are completely unknown, we feel like it should be protective for compounds that are known. But there's been some pushback and some discussion on that point. So you can see I've copied and pasted some feedback that we've received from the FDA on this matter. And we've been having this ongoing conversation. So one of the things that we hear from the FDA is whatever evidence is there that supports that those compounds aren't on the TTC exclusion list. And you can see on the right-hand side of the slide, within ISO 21726, there's a list of compound classes that the TTC really shouldn't apply to. And I think that this bit of feedback from the FDA is a valid point. it's clear that compounds in this class, these aflatoxin-like compounds and N-nitroso compounds are not covered by the TTC, and we ought to take a look at those that are above the AET and below the TTC and determine or assess whether or not they fit into one of these categories. And that's something that we can do. For the other compounds, what we've heard is something like this, so in various different formats. The FDA will say, for several compounds, you state that the exposure is below an appropriate TTC. However, use of TTC is recommended for toxicological risk assessment of each identified compound for which there is no toxicity data available. I would also say for unknowns. Therefore, the TTC limits should not be applied to compounds for which there is toxicological data available. Please revise the toxicological risk assessment to include use of TTC only for compounds in which there is no adequate toxicological data available. Now, if you've ever done a toxicological risk assessment, or if you've contracted with somebody to do an assessment of extractables results, the burden and time, and I would also say cost, of that evaluation scales with the number of compounds that need to be evaluated. Sometimes you might have many compounds below the AET or below the TTC but above the AET, let's say 50. And if the cost structure, if it takes 6 hours per compound to do proper research on all of those, that really has drawn out the timeline involved with the evaluation of that. And so we want to kind of resist this idea in a sense that it dramatically increases the burden of doing a toxicological risk assessment with perhaps questionable value when it comes to patient safety and being conservative. And so really that was what I wanted to do in this poster presentation, is try to answer that question. So is it true that by doing a literature search and deriving a tolerable exposure per ISO 10993-17, that would make an assessment more conservative, or sometimes even produce thresholds that are lower than the TTC that was applied? And we've done this on a case-by-case basis. You can see an example here where the FDA had a concern about 2 particular compounds. We went back and made this evaluation where we derived a TE. So you can see that both compounds were below the TTC of 1.5, but above the AET. The FDA didn't like that we applied the TTC to that. When we did this literature research and looked for structural alerts and all these other things, we were able to derive a tolerable exposure. And you can see in both cases that it's significantly higher than the TTC. And the way that this process of generating a tolerable exposure goes is essentially what needs to happen is the literature needs to be searched and a no observed adverse effect level is identified from an experimental toxicological study in animals. And that NOEL, the no observed adverse effect level, is extrapolated to humans by using a series of uncertainty factors. In 10993-17, there's a set of 3 of these. One is for interspecies variation and then intraspecies variation, and then the third is related to data quality and extrapolation due to different routes of exposure. Once you have a tolerable intake, that's the TI, you make that into a tolerable exposure by taking that amount, which is in units of micrograms per kilogram per day, multiplying it by a standard body weight for the population involved, and then also an uncertainty factor, a utilization factor that scales for the number of concurrent exposures to that type of compound that might happen in a hospital setting. And the default factor for the utilization factor is 0.2. So it's basically assuming that there would be 5 parallel sources of compound in an exposure setting. So to see if this is true in general, that the TTC is more conservative or produces a lower threshold than a derived TE, I went back through our most recent data and I identified as many compounds as I could that were in this range, below the TTC but above the AET. And in the last few months, there were 180 compounds that fit that criteria. Of those 180, 130 of them were unique, so there were some duplicates across studies. And then for each of those 130 compounds, I searched the literature to identify if there was any toxicological information available. Of those 130, there were only 60 compounds for which experimental tox data was available. And you can see here, I plotted just the solubility and molecular weight of those compounds just to show that there's a range of properties here. So we span molecular weights going from about 50 all the way up to 600 grams per mole, and the solubility is all over the chart. So this is really a random selection of compounds that were observed from medical device extracts in this concentration range that were available. So for those 60 compounds with toxicological information, I went back and found a NOEL for each of them and then computed a tolerable exposure per the formula in ISO 10993-17. So all of the NOEL data that I found, this experimental tox data, was in the form of oral exposures to rats. So the most clear connection, if we were going to apply that data to an actual use scenario, would be if the use scenario also was like an oral exposure. And a case where that happens very commonly is in gas pathway medical devices where water can condense inside the gas path, and it's possible that the user of the device might swallow some of that condensed water. And so that was what I was kind of imagining here. So if we take that the experimental tox data in the literature and extrapolate it for permanent oral exposure in humans, then you get this plot of tolerable exposures that I show in the graph. You can see different patient populations are broken out there. So oral exposure in adults, pediatrics, and neonates. Neonates being obviously the most vulnerable population with the lowest body weight. And you can see that in the lowest case, the TE is roughly 100, which is far, far above the threshold of toxicological concern of 1.5 micrograms per day. So for something like oral exposure, that means that there really isn't an identifiable case where the TTC is less conservative or less safe than deriving a tolerable exposure per the information in the literature. And really the point that we want to make here is that we can go through this effort of researching the literature and deriving a tolerable exposure, but it's still worst case to use a threshold of toxicological concern when we can. So you might say that, well, there are a lot of medical devices or exposure situations where it's a lot more invasive than just oral exposure. So I thought of that scenario as well. So I took that same body of toxicological data and then applied it to the case of permanent exposure into the vascular system. And you can see, so something like a drug that's administered via IV, or you can think of a device like a stent. And even in the case when it's permanent exposure directly to the blood in a neonate, the lowest TE out of the entire set, spanning these 180 compounds that fit inside this concentration range, the lowest TE was 9.8 micrograms per day. So that's still significantly higher than the TTC of 1.5 micrograms per day. So I think that in conclusion here that we can say that we looked at 180 instances of compounds that were above the AET but below the TTC, and we asked the question, okay, is using the TTC the most protective of patient safety? And after we went through this process, many of the compounds didn't have any toxicological data at all, and there the TTC is explicitly justified. So in many of those cases, that using the TTC isn't questionable. In 60 of those cases, there was toxicological data, and we were unable to identify any scenario where the TTC would be higher or less conservative than a tolerable exposure derived on the literature. So with that, I'll go ahead and close. I just want to mention that there's my contact information there. I'd be happy to talk about this with anybody that might be interested in having a further conversation by email or phone. And then finally, we have a whole lot of these types of resources. There's tons of stuff going on in the world of biocompatibility and sterilization and extractables and leachables. Please feel free to visit the Nielsen Labs and Sterigenics websites if you're interested in learning more on any of those topics.